Hello friends. Welcome back. Today we are going to cover an interesting topic, plague. Its historical impact, etiology, clinical features, and management. Plague is a severe infectious disease caused by the bacterium Yersinia pestis that can cause peripheral gangrene, severe pneumonia and lymph node enlargement called buboes. You can also see in this picture plague bacilli having a safety pin appearance on gram stain. Plague is not commonly seen in the modern world, but it remains an important bioterrorism agent. That is why it is important to know everything about this deadly disease. First documented pandemic caused by plague was the Justinian plague that occurred between AD 541 to 549. The bacteria arrived in Rome and Egypt in 541. It spread around the Mediterranean Sea until 544, and persisted in northern Europe and the Arabian Peninsula until 549. This pandemic killed 20% of the population in this region. The next major pandemic is also named the Black Death. It lasted from 1346 to 1353. This was a bubonic plague pandemic that affected whole of Europe. 50% of Europe's population was killed in the Black Death pandemic. It was described in Latin as pestilentia, pestilence, epidemia, epidemic, mortalitas, mortality. The words like mortality and epidemic are byproducts of the Black Death. In this picture you can see the strange costume of medieval plague doctors. The causative organism of the Black Death was not known. It was believed that good smell will cancel out the disease, so people used to walk with a flower under their nose. The beak of the plague doctor was filled with herbs, spices or lavender. Third pandemic of plague occurred between 1855 to 1859. It started in China and spread to all continents. This pandemic killed 10 million people in India alone and 15 million worldwide. The causative agent of plague is Yersinia pestis, a gram-negative, rod-shaped coccobacillus. Transmission of plague. Plague is a zoonotic disease. It is seen in restricted geographical regions. Over 200 species of animals are definitive hosts of plague that mainly include rodents. Humans can get infected through flea bites that leave dead rodents and search for new hosts for blood meal. Humans can also get infected by consuming infected meat. But the pandemic form of plague that involves human-to-human transmission occurs through inhalation of respiratory droplets. Plague can manifest in various forms. First is bubonic plague. This is the most common form, characterized by swollen lymph nodes also called buboes. Patients develop sudden onset of fever, headache, chills, and swollen, painful lymph nodes. Second form is septicemic plague, in which plague bacteria multiply in the bloodstream. Patients develop fever, chills, extreme weakness, abdominal pain, and finally shock and bleeding into the skin and other organs. They can also develop peripheral gangrene. The third form of plague is pneumonic plague. It manifests by involvement of the lungs, can be primary or secondary, fever, headache, weakness, rapidly developing pneumonia, shortness of breath, chest pain, cough, and sometimes bloody or watery sputum. Diagnosis, based on careful history taking, symptoms and clinical examination. Since in the modern world, we do not see patients of plague, it is easy to miss frank cases of plague. High fever, peripheral gangrene, buboes, pneumonia, hemoptysis and occurrence of many similar cases originating from a restricted area are the pointers towards a plague epidemic. Lab diagnosis of plague. For bubonic plague, send pus or fluid aspirate to the lab for staining and culture. For pneumonic and septicemic plague, send sputum and blood. The transport media used for plague is Carey-Blair medium. Gram stain of the sample shows pus cells and gram-negative oval coccobacilli with rounded ends that can be surrounded by a capsule. In this picture under direct microscopy you can see bipolar or safety pin appearance, ends are dark, center is clear. This appearance is seen with Wiesen stain or methylene blue staining. Plague bacillus grows on ordinary media. Yersinia pestis, the causative organism for plague, is aerobic and facultatively anaerobic. It develops a capsule at 37 degrees Celsius. On blood agar, Yersinia pestis develops non-hemolytic, dark brown pigmented colonies. On macaque agar, it develops non-lactose fermenter colonies. When grown in nutrient broth, it shows granular turbidity with surface pellicles that are formed later. When grown in nutrient broth with oil floated on top, it develops characteristic stalactites-like growth as seen in the picture. We can also grow plague bacilli and Yersinia specifics in agar. SYN stands for Spsalodin, Ergason, and Novobiocin. It is a selective medium for plague. It is used for culture of sputum that contains normal flora that can outgrow plague bacilli in the non-selective media. When grown on blood agar, plague bacilli develop phrygid appearance at 96 hours. When we have obtained a growth on culture, we do a gram stain. Plague bacilli are seen as pleomorphic coccoid, coccobacillary, bacillary, filamentous and giant forms. They are non-motile at 25 degrees Celsius and 37 degrees Celsius. Next, we test the biochemical reactions. Yersinia pestis is glucose, mannitol, and maltose fermenter with no gas production. It is also catalase positive and oxidase negative. Other biochemical reactions include indole negative, urease negative and citrate negative. It is methyl red positive and Voges-Proskauer test negative. In emergency settings, we can use rapid diagnostic tests for plague. RDTs use detection of Yersinia pestis antigen and can be done on the aspirate from buboes or sputum. The underlying principle of these RDTs is direct immunofluorescence, ELISA, or immunochromatography. Other serological tests for detection of antibodies include passive hemagglutination test, complement fixation test, and ELISA. They have limited value in diagnosis because of their late appearance, and antibodies can remain positive for several years, but serology is important for epidemiology. Plague can also be diagnosed using molecular tests like PCR, to gene-coding F1 antigen, pesticin gene, and plasminogen activator gene. The treatment of plague involves administration of antibiotics. We can use streptomycin, gentamicin, doxycycline, or ciprofloxacin. Patients may also need supportive care with intravenous fluids, oxygen therapy, and respiratory support if needed. Pneumonic plague is contagious, and therefore isolation measures may be necessary for pneumonic plague to prevent spread. Prevention measures include control of cases by early diagnosis, isolation and treatment. Control of fleas using insecticides like DDT or BHC, and control of rodents. All close contacts of pneumonic plague patients must be given chemoprophylaxis. Doxycycline is given 100 mg but for 7 days. Regarding plague vaccine, it is available and used only for prevention of anticipated outbreak. It is a formalin-killed vaccine prepared in Hafkin Institute, Mumbai. 2 doses are given subcutaneously, 4 weeks apart, and a booster after 6 months. It is not protective against pneumonic plague and can have considerable side effects. This map shows the current endemic areas in the modern world that include Africa, Asia, and the Americas. Plague remains a potential bioterrorism concern because of its potential to be used as a bioweapon. To conclude, plague is still endemic in many regions and outbreaks occur in cyclical fashion. Healthcare workers must be trained to identify the outbreaks early. Rodent and flea populations must be kept in check in urban areas. Stock of antibiotics and PPE kits must be maintained in endemic areas. Rapid diagnostic tests must be made available for quick diagnosis in ED. So, this was all about plague. Hope you liked it. For more of such videos on infectious diseases, like, subscribe, comment and share.